Well, thank you, Chris. And actually, I feel encouraged by what you've said, because you say you're using it, because I do know on this subject that the community is somewhat divided, that there are some of us who have integrated therapeutic drug monitoring into our routine clinical practice, and there are others, many who are sitting in this room, who've told me that I'm wasting my time. This is completely redundant in the era of modern drugs that we have. Why are you doing it? So I guess one opportunity, because I'm incredibly honored to be giving this talk, is to try and stimulate discussion about where we should be going with this potentially powerful tool to leverage and improve outcomes in dermatology. And of course, what I think is immutable is that for a drug to have action, you need the drug at the site of the target. So that is without question. But the idea that the right dose is crucial, that's also pretty established. And in fact, Paracelsus was the father of clinical pharmacology, who really established the idea that either too much or too little drug could cause harm, and that getting the right dose was crucial. And actually, in common with perhaps me standing here in 2023, Paracelsus couldn't get his ideas taken up by the general community, and was said to be a very difficult man. I don't think I'm difficult, but he was. I think this definition is helpful. It's broad. And it encompasses all the aspects of therapeutic drug monitoring. So it's about individually adjusting the dose to improve outcomes in general, or of relevance to us here, maybe special populations. And what it involves is information either before you give the drug or after. And that after information may include measurement of drug levels, which is what we associate with therapeutic drug monitoring, but not always. It might be PK monitoring, but it might also be PD monitoring. So everyone's familiar with warfarin. I mean, an old drug, but everybody's measured, I'm sure, as juniors, the clinicians in the room measuring INR to adjust the dose of warfarin. And in fact, if you canvas dermatologists, there was a big survey done recently led by Joe Lambert. Around 70% of dermatologists are adjusting the dose of biologics to improve outcomes. So we are all doing it. But you could argue that with implementation and further development of more sophisticated forms of therapeutic drug monitoring, we could do a whole lot better. So what I thought I'd just cover in the next few minutes is what's the evidence to support TDM, and I'm going to talk primarily, because we're a psoriasis audience, about psoriasis, drawing on information from the other immune-mediated diseases. And I am going to talk mainly or completely about biologics, although the principles and challenges are also generalizable to small molecules, although the PK of biologics are very, very different to small molecules, and I'll talk about that. And then I'm going to talk about a few practical aspects, and maybe we could have some discussion about what the opportunity might be looking forward. So just a note about the PK. I mean, the pharmacokinetics of biologics is peculiar and specific in that they are large molecules. And very, very different from small molecules, at least the biologics that we're using at the moment. And so the bioavailability of drug is a fraction of that administered, particularly subcutaneously. And so thinking about the principles of ADME, absorption depends on injection technique, local factors, and the degree to which you have pre-systemic clearance. And that's probably one of the reasons why obesity is such a challenge in getting adequate drug exposure in the psoriasis population. In terms of distribution, disease activity may be important around this idea of antigen sink, and perhaps, and this has not been well investigated, disease phenotype and site. So if you look at drug development and pharmacokinetic investigation and modeling, there's relatively little done on the distribution of the drug. So it might be one of the reasons palmoplantar psoriasis is so challenging therapeutically as it may be to do with drug exposure at that site. There may be something specific about that. And then clearance, of course, we're all unfamiliar with the ADA story, but again, antigen mass disease activity may be important. And the FcRn-mediated salvage pathway is crucial for the recycling of biologics. And in terms of PK/PD models, parameters that have been identified, depending on the drug and the model, but weight is a key one in terms of influencing drug exposure, but also sex, age, ethnicity, diabetes, in some models, high sensitivity CRP and ADA. But I suppose the key point is that it's complicated, it's complex, and there are multiple factors influencing drug exposure at the site of action, and these change over time according to those variables and also disease activity. But what is certain is that the relationship between drug exposure and outcome, one of the key requirements for therapeutic drug monitoring, is very well established for all our biologic therapies. It's part of regulatory approval. So the data is there. So this is a typical, I'm not going to go through all of this detail, but I just wanted to point out a couple of points. If you look on the left side, this is the modelling that was done for risankizumab, and I forgot my glasses, so I can't read this. But anyway, the key thing here, for those who don't maybe look at these, is the black bar and points relate to the observed drug exposure, and the blue is the model. And on the left-hand side, I think it's interesting that thank you very much for it, that the degree of variability, well, the point is it depends on your outcome, but if you look in the top left-hand corner there, PASI 75, there's a much less variability compared to, say, the bottom right for a PASI 100. That's one point. And the second point I wanted to make is this plot on the right-hand side. There is, if you look on the y-axis, the observed drug exposure, they've looked at trough concentration. And what struck me as this plot, say, if you take a single body weight 75, the range of measurable drug level for any weight. So it's just to make the point, there is huge between person variation in the amount of drug that you can see. And the other point is that there is a marked plateau of drug. If you look at, for example, risankizumab, but it's honestly the same for all the modern, P19s, the P17s, in that there are a lot of patients there who have more drug than's required in order to achieve their outcome. And that, of course, is being done to ensure that the population exposed to that drug have got the highest probability you're going to capture response in your population. And based on the assumption that these biologic drugs are actually pretty safe. So pushing the drug to a high dose doesn't carry with it any great risk, and that which is borne out by the data. These drugs are incredibly safe. So we know there's a relationship between drug exposure and outcome. We know that there's a lot of variability between patients, and we also know that there is variability in response, particularly if we're going for clear skin in those very hard endpoints. Patients want clear skin. Now, approaches to therapeutic, so those are all good sort of important features to justify thinking about TDM. There are two approaches: reactive, in other words, reactive to the clinical status. And you could argue, and actually the evidence suggests, that there's not much benefit really over clinical observation. Patients relapsing, you switch the drug. It's useful to know if they've got anti-drug antibodies or if they're not taking their drug. But if you look at the trials, in general, reactive TDM, the data isn't so convincing. The idea of proactive therapeutic drug monitoring is going after a particular PK target, or it might be a PD biomarker if you have one. And in IBD, they combine the two, PK with fecal calprotectin, for example, to make the models even more accurate. And it can be done either induction or remission. And the evidence that's there suggests that actually the need for TTM during induction is probably not necessary, particularly with the modern drugs, because the dosing is so aggressive during the induction phase. But there might be an opportunity in remission. Now, evidence, I'm not going to go through it at all, but the take-home message is that TDM has been most widely adopted in inflammatory bowel disease and by gastroenterologists, and even in that area, most of the data relates to TNF antagonists. It's relatively low quality, actually, although it's very widely used. And there's a gap between what is incredibly sophisticated, well-published data on the drug exposure outcome relationships, mostly sitting in industry, over 45 trials studied, published in the last 8 years, moving to evidence for the clinical utility of TDM, and the data is mainly cross-sectional, retrospective, or relatively small prospective studies. This is just one summary, and in fact, I'm involved in a systematic review exercise across all of more unupdated one. But the thing I wanted to point out here is the evidence for benefit, and this is in IBD using TNF antagonists. So I know there are many people sitting in the room saying, I don't use them anyway. But it does favour proactive TDM, and this is primarily during the maintenance phase. In other words, to try and maintain remission. The one randomized controlled trial there, you'll see 122 by Van der Kasteel, actually the population in that trial were already dose optimized before they were randomized to the maintenance dose. So they already measured it was with infliximab, they already knew that they were in the ideal therapeutic range before they randomized. So that study has been tricky to interpret. But a very important study that was published in 2021 by the Norwegian group, this is the Nordrum study, they did a randomized controlled trial across immune-mediated diseases, including patients with psoriasis, and showed that during maintenance, disease control was better, there were fewer flares, and they, as a consequence of that, in fact, it's triggered rapid recommendations around this. And this is the subgrouping of the different diseases. The problem for us with the therapeutics that we use is that the PK TDM targets are not available for most of the drugs that we use. We've been involved in developing them for adalimumab and ustekinumab, and Jo Lambert, who's done a lot of work in this area too, she's working on and got data on gazarkimab and ICSI, and is developing that in those other drugs. But there's still relatively little evidence around the PK target. And the other problem with some of the studies is that, certainly from the IBD literature, the endpoint that you're aiming for is very, very important. So if you're looking at biologic, this is in relation to infliximab, but there's no reason why this isn't generalizable to all drugs. The endpoint determines how much drug you need. So if you're looking for biological remission, that's 2.8, that's clinical remission, versus endoscopic goes up to 4.8, versus histological 10.2. So we need to think about what our endpoint is in developing TDM. And the other challenge with the current approach is that even with a therapeutic range, within that therapeutic range, not all people are exactly the same. There'll be variability. And therefore, we need to be thinking about individual drug targets for a particular individual. And this is where the kind of discipline is moving to these more sophisticated model-informed individualized Bayesian TDM, where you have your population-based PD model, a priori information that you might put in relation to your patients. And Michael mentioned earlier, the data around the HLA DRB1. It might well be you could put in other genetic information, and then you run the model and you estimate as far as you can based mainly on the population for that patient. But then you input their PK, and the advantage of this model is it doesn't need to be trough, because you can take into account the time and their PD, their individual response, to really drive much more precision dosing. And these sorts of dashboards are being tested and trialed now in IBD, and there's opportunity probably for some subpopulation high-need patients in our discipline. To talk about the practical aspects related to TDM, they're massive. They're massive. First of all, assaying the assays, at the moment anyway, access to assays and harmonization, standardization, and there are efforts across Europe to try and do this, but this is still very fragmented to actually get the test. And then getting the results in a timely, useful turnaround, testing the drug level and then amending the drug to do that. And there are strategies around home sampling to try and expand point-of-care testing. And then most importantly, what do you do with the results? So there's a dearth of well-evidenced, high-quality TDM algorithms, and I'm involved in this effort for recommendations around proactive TDM, partly driven by these new Nordrum studies and emergent data, and it's in progress. It's very interesting. It's not rapid, as with all guidelines. It's going to take us time. So we tried to develop this just locally. We developed an algorithm based for adalimumab, based on our data and others that have been published, and then implemented into the clinic. And this paper that we published recently, I think, is useful as a kind of proof of principle, not necessarily about TDM, but how do you introduce new tests? How do you do it? We've got this emergent ambition, or not an ambition for stratified medicine and biomarkers, but the challenge is getting it into the clinic. So you validate the test, decide who you're going to test, develop the protocol, skill everybody up, and then evaluate how effective and whether it's worthwhile. And I think this is where maybe there are, even if you aren't an advocate for TDM, and I am, but even if you aren't, the idea of precision dosing and getting that framework right is really a framework for precision medicine more generally. And these principles are also generalizable. We need to really have strong industry academic partnerships to move that data into around PK/PD in this context through to algorithms that are fit for therapeutic drug monitoring to decide the target and so on, and the population. We need implementation tools. We need to partner with diagnostics companies. And we also need some well-designed trials in the relevant target population. In other words, where do we see this tool being of most value going forward in the context of some of the modern drugs that we've got? And is there an opportunity to more intelligently minimize drug exposure in well-controlled disease? So as mentioned earlier, that with the P19, people come off drug and they're in remission off drug for a while, and that is true, but there are also people who relapse. And we do know that those people who have disease well controlled, many of them have excess drug. And so this is one of the things that we're trying to address in one of our trials. We're taking a population of people who've got very well controlled disease and randomizing to continuous, or as they need it, when their psoriasis pops back, they're going to re-inject themselves immediately. I mean, the threshold for restarting in the trials is often quite high, but we're going to leave it to patients to decide exactly when to start. And then the third arm is around a model-informed precision dosing to try and minimize their drug exposure to maintain well-controlled disease. But in designing this trial, what do we mean by well-controlled disease, and how do you capture it over time? A lot of the endpoints are PASI 100, PASI 90, but that's at the end of the trial. And thinking about that, you have to think about, well, what are the thresholds for low and high activity? How do you define a flare? There's really no consensus, as far as I know, across the community. So in summary and looking forward, there is fairly good evidence that proactive TDM during maintenance improves outcomes for infliximab and adalimumab, low quality for the others. Barriers to implementation are multiple. Model-informed TDM, I think, does hold promise, I'm excited about it, but we need some consensus amongst the community about terms and endpoints. You know, IBD community, what do they need by remission? You know, across immune-mediated diseases would be helpful. And the framework that we might develop has value well beyond TDM. Thank you very much.
Thank you very much. Yes.
Okay, so
This is the man in the room.
I know. Yeah. So first of all, I really think you need to separate out IL-23 blockers from all other MOAs with psoriasis, because there, when drug is not in the system, you still have a PD effect, right?
Of course,
So
yes.
with the other drugs, it's more closely linked. It's really hard to spread those out. So in your last study, you mentioned PRN dosing. I think it's perfect for 23 blockade and not good for any other MOA. So is that 23?
The proof of principle is in P19.
Okay, great. The other thing I want to say is just drug companies are so obsessed with PK, you know, and they're trying to figure out the initial dose. And for 23 blockers, it doesn't work that way because the effect lasts so long when the drug's gone. There is more of an effect with high dose versus low dose. So I reported at the World Congress that we had 83% PASI 100 with high dose risen, and it's it's from 50 to you get more PASI 100, right, with more drug. And the IBD folks use 600 milligrams with no problem. Something I've been told 1,200 milligrams, no side effects. So why in dermatology do we use 150 milligrams when this other community uses 600, 1,200 milligrams of resin? So I think that 23 blockers should be treated separately in your work. Like, it's almost like the PK relationships, the spreading out compared to other MOAs.
I suppose, I mean, actually what you said means that you're a great advocate of TDM, really. You because, you're absolutely right. But your assumption is that the PK/PD is linearly, it's a correlation. But actually, with the PK/PD, it models, it takes into account that delayed action. And except you've just said, Andy, that yes, but that is related to PK. What you're absolutely right. The level of drug that you measure in the blood may well not indicate what's in the tissue, but that's all to do with PK. So we're all talking about the same thing. So, but actually, you're right. I mean, in IBD though, it is a different situation. So we know in IBD that there's huge amounts of drug being lost through the gut. Actually, there's a huge gut antigen sink and loss in the gut, which is one of the reasons why in induction they have to use such very high doses. So that's one of the things, and why. But the idea of giving IV, I agree. But I would argue that we probably need to measure the drug levels in the tissue to better understand the distribution, which was my what. Yeah. So I think we're agreed, Andy.
Okay. One more question. Yeah, Johan.
Yeah, I have a quick question. I hope. Do you have any concerns about this? That you increase the risk of anti-drug antibodies with intermittent dosing, or that you have more chance of disease escape?
Yeah, actually, well, that's a really, really good point, because actually one of the...
The question was about the risk of anti-drug antibodies if you treat as needed.
Yeah, I mean, one of the... so one of the ideas behind this maintenance proactive therapy with infliximab, one of the reasons it's felt that, if you it's by maintaining that chronic drug exposure that you do minimize anti-drug antibodies. So, and of course there was a famous study where, it was randomized and there was more immune reactions. I think that those more antigenic, immunogenic drugs, intermittent therapy may well be more challenging, but the immunogenicity of the new drugs is much less. But you're right. I mean, it's something we need to watch. Yeah.
Okay. Yes, I think that's it. Interesting. So a patient example where you would take a PK value and use it.
Well, I mean, in our practice,
Is
we
in
are
the
doing,
practice.
at the moment, we're doing proactive PK in adalimumab.
In
and
adalimumab,
in
in
infliximab.
psoriasis patients?
Yes,
Or,
yes,
okay.
yes. And actually, it's surprising how often it's helpful.
It's
I mean,
helpful.
yes. I mean, in about 20% of our cohort, we made adjustments that we wouldn't otherwise have done without a PK. Yeah.
Okay, good.
I mean, the other thing about the PK is whatever your drug level is today, it predicts your clinical status in about 6 months' time.
Yes.
So it has predictive value. So you're not chasing the disease, you're ahead of it. That's the advantage.